Hello everyone, my name is Rahul. In the previous video, we had discussed about the use of genetically modified organisms. In this video, we are going to discuss about resistance against pest, that is pest resistant crops, which are a type of genetically modified crops. Firstly, we are going to discuss about Bt cotton. The term Bt stands for Bacillus thuringiensis. Bacillus thuringiensis. This bacillus produces toxic crystals. They produce toxic crystals of insecticidal protein during their growth. These insecticidal proteins, they have the ability to kill certain insects, such as lepidopterans, which include tobacco budworm, armyworm, etc. Then coleopterans, which include beetles, and dipterans, such as flies, mosquitoes, etc. Now, this ability to kill the insects comes due to the presence of a protein known as cry protein. This cry protein is toxic to the larva of insects. Now, there are various categories or various types of cry proteins, and each protein is specific, and each protein kills only a certain group of insects. Now, this cry protein, it is a product of a gene called as cry gene. Now, please note the difference that when we write the name of the protein, C will be capital, and when we will write the name of the gene, the C will be small. So, this protein is the product of a gene called as cry gene. This cry gene can be separated. This cry gene is separated and introduced into host plants. This cry gene is separated and introduced into the host plants. So what this will do is, it will lead to production of cry protein in the host plant. This cry protein, it is secreted or produced by host plant in inactivated form. Now, when the insect will feed on the plant, 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 feed on the plant this protein will reach the gut of the insect and gets activated due to alkaline pH. This inactive protein requires alkaline pH to get activated, therefore it will not harm the humans, because in humans the environment in the stomach is acidic, the pH is acidic, so this protein will be completely inactivated. This will lead to lysis of cells in the gut and ultimately the death of insect. So the insect feeding on this plant, which is containing this cry gene, will die off. So BT cotton is a good example of this type of plant. So lysis of cells, along with there is swelling, pore formation, and lysis of cells of the gut of the insect, and leads to death of the insect. Now the proteins encoded by various cry genes are different. Example, from the gene cry1AC, the protein that is produced, it controls cotton bollworms. So the gene cry1AC will produce a protein that will control the cotton bollworms. Similar is the case for cry2AB. It also controls the cotton bollworms, or rather same. Cry1AB, it controls corn borers. So different varieties of different genes, they will produce different proteins which are specific for different types of insects. Now, apart from BT cotton, other crops such as BT tomato, BT brinjal, BT corn are also available. They all work on the same principle. Now earlier what we used to do was, we purified this protein from the animals. We took up the crystals from the we took up these crystals from this bacterium, purified it, and produced it into sprays, and we used to spray these crystals on the plants. Now the disadvantage was, because we are spraying, many a times the spray would not be completely performed, and a lot of product was going to waste, and sometimes the product was not even present on the plant. So to prevent all this, to prevent losses, what we did was, we used biotechnology to introduce these genes into the plant itself, leading to production of this cry protein, and ultimately which kills the insect, making the plant resistant to these pests. So with this we finish off this video on BT cotton. See you in next video.